So this idea that if the microbe is swimming around, maybe in a stream or something, or in a test tube, it will, and assuming it has glucose, it'll be running this to pyruvate, and then it will be assessing its environment. And if they're smart, they're looking for oxygen. Because oxygen is the best electron acceptor on the face of the earth, which means you'll make more ATP using oxygen than any other strategy. So if E. coli is swimming around in that broth tube that we give you and it's like, oh yeah, there's plenty of oxygen, then they'll take that pyruvate and go into the citric acid cycle. And when you do that, when the microbe is doing that, what are the payoffs for the citric acid cycle? What's one good thing the microbe gets from this?
Electron transporters.
Yes. And those would be NADPH or NADH, FADH2. So in my head, I equate those to Tinkerbell. And the electrons they're carrying is the pixie dust, because that's kind of what they like to do. They like to fly around go share those electrons, go sprinkle some electrons into other reaction pathways. And they're really good at that. So that's good. What else is good about the citric acid cycle for the microbe? Yes.
More CO2.
Yes, they're breaking down. So if you think about glucose that started off in glycolysis, they say by the time the citric acid cycles run, you've taken that glucose and completely broken it down to CO2. And that's also a good thing because you get more energy in the end by completely breaking down that glucose, that carbon molecule. So that's good. What else? There's one more key thing that the microbe is getting. Yep, ATP or GTP. So we still haven't finished the thought about oxygen because that's not here yet. All right, so now we got to think about, this is kind of like the middle step. But the microbe is still going, yeah, there's still oxygen. So then they can take those electron carriers. Remember, they go by many names. It depends on which textbook and which reading or who's talking. No, not that. Where am I going? Where is it? There, Tinkerbell. So those can go called many things. But basically, now they're going to float over to the electron transport chain process. And in that, they'll be able to donate their electrons. So that's what we're looking at here. So you see the electron flow. Here you have NADH and then FADH2 coming in at complex 1 and complex 2 respectively, donating their electrons through this chain, series of donors and acceptors on down the chain until you see oxygen as the terminal or final electron acceptor. So that's what the microbe is hoping to do. They're hoping to run something very similar to this where they have oxygen as the terminal electron acceptor, because when you have electrons flow like this through the chain, it's linked to proton pumping. And so it's really a mechanism for the microbe to generate a proton gradient. And so anytime you think about electron flow, you need to think about a proton gradient or proton pumping across the membrane that leads to a proton gradient. And I showed that analogy of the people, the sledding downhill with the dog jumping onto the sled and the dog representing the proton, how the dog takes advantage of that potential energy and jumps on the sled as it starts to go down the hill and then can jump off at the bottom. And so that's kind of like the proton moving across the membrane. And once you have a proton gradient across a membrane, in this case, it's the cell membrane, but it could be a mitochondrial membrane in your body right now, then the protons want to come back through. They can't get through the membrane itself, so they have to do the ATP synthase. And then they get a lot of ATP. Whoops, not that one, this one. So here you can see 34 ATP from having the electron transport chain process. Well, that's the big payoff. Glycolysis, you got a net of 2 TCA cycle, not much better. And then you get a lot with the electron transport chain with oxygen as the terminal electron acceptor. Okay, so these questions I'm asking you right now, think about these questions, what we just went through. Okay, that's kind of the concepts I'm asking you to know for the exam, not the nitty gritty. Don't get lost in the detail. It's not a biochemistry class. You don't have to know every single step of glycolysis. But the big picture ideas, what is it the microbe gains from this? Why are they doing it? Does that make sense? Okay, so don't get lost in the details. Question, yeah,
Is the exam gonna be next week or the week before break?
I think it's the,
Cause right now it says it's the week of the 16th, which is, I believe, next week.
is it next week? Yeah, I think it is next week, because then you have a lab exam right before break on Thursday. And I don't put the lecture and lab on the same week. I don't want to drive you all nuts. So yes, the exam's next week. Thank you for reminding me. So it'll probably be Friday again. I'm guessing that's how it's going to work out. Questions on these ideas of glycolysis, TCA, or citric acid cycle, electron transfer chain? So I often envision the microbes as kind of like, I used to rent a 2-bedroom duplex that had 2 bedrooms, and my favorite thing about that whole, well, 2 favorite things, it had a walk-in closet, never had one of those before, and it had a real pantry, a walk-in pantry. And so I often envision like a microbe is going into a pantry and slinging open the doors and going, what do I got to cook with? You know, what can I eat? And this is kind of what they do on a minute by minute basis. They're like, do I have oxygen? Yes, okay, I'm gonna do all this. And if the answer is no, then we gotta talk about what they might do instead, okay? But it's pretty much, they're just always assessing their habitat. What do I have to work with? What kind of creation can I make? I'm not a chef. I always envy people who can just grab stuff out of the pantry and make it happen, but I can follow a recipe. I'm not a creative chef. But anyways, so let's think about some other ingredients a microbe might have and some limitations it might have to deal with. All right. So then we started thinking about the redox tower and how they can do these redox reactions. And again, it's all a matter of what are they surrounded by? What is their habitat giving them? What do they have to work with? And so we talked about this last time, how we have these redox couples in this so-called electron tower, and how the reduction potential, they always do it very negative at the top, and then approaches 0, and then positive, and the most positive is down at the very bottom. And oxygen is always at the bottom because oxygen is the best electron acceptor. And so whatever's to the left of the slash line, that is the one that will be able to accept electrons, is ready to accept electrons. It's in its oxidized state, so it's ready to accept. If it's to the right of the slash, that means it's ready to donate electrons. Okay, so H2, hydrogen gas, is a really good electron donor. And so here, when it's written down at the bottom, O2/water, that means oxygen is like really good at accepting electrons. It is really greedy for electrons. And it means that glucose, on the other hand, is really good at giving off electrons. So is hydrogen, so is the sulfate. All right, so then these arrows that you see here are showing potential reactions and how to think about them. So here we have the black arrow. If you look at the number 1, they've written it here. So if you have hydrogen and then you have fluoride and succinate, well, you know fluoride's to the left of the slash, so that's going to be accepting electrons. We know hydrogen is really good at donating electrons, so that's the reaction, and then you end up with succinate, which is to the right of the slash, right? And if you calculate the free energy for that, delta G, it's negative 86 kilojoules. That's pretty good. By the way, we'll get to it in a future slide here, but what I want you to remember is negative 32 kilojoules. Negative 32, that's the magic number. If you can generate at least negative 32 kilojoules in a reaction, that means you can make ATP. And that's what you gotta do. So you know negative 86, that's even better. So you can make at least a couple ATP off that. All right. The red arrow goes from the hydrogen couple down to some nitrogen there. So we know that nitrate, NO3 minus is nitrate, that's to the left of the slash, that's going to be accepting electrons. We know hydrogen is really good at donating, so there we go, hydrogen plus nitrate, and then you'll get the nitrite, NO2 minus, on the right of the slash, and then water. Free energy is even better, negative 163, so now you can make even more ATP than reaction 1. And then the blue arrow goes from hydrogen, again, donating electrons, oxygen accepting, you yield the product of water, and the free energy is the best. So the way to think about this, I have it in the notes as well in one of these here, is that the farther the electron falls, so to speak, down the tower, the farther it falls, the more free energy you're going to get from that. So that's why hydrogen donating electrons and falling all the way down to the bottom of the tower gets you the most free energy, which means the most ATP. So the farther the electrons go down the tower, the better. Right? Okay. So here you can see, and this isn't a complete tower, of course, because there's all kinds of molecules out there. But take note of there's an option down here of NO3 minus nitrate. NO3 minus. We'll come back to that. Questions on this electron tower? Somewhat get it? I'll go over it again. But this takes some, well, if you're like me, when I first learned this, it's like, what? I had to look at this seriously more than once. So please spend some time with it. We'll come back to it on Wednesday. All right. So one of the goals the microbes are trying to do is to get some ATP so they can stay alive. They can do all kinds of activities. Maybe they want to move their flagella. Maybe they want to start reproducing themselves. Maybe they want to build some biomass. All right. So ATP is the main one, but there's others. I'll show you a picture too, but basically the ATP has a lot of bang for the buck because of the anhydride bonds. When those get cleaved, it releases some energy, so that's good. So ATP can be very useful for any living organism, as well as phosphoenolpyruvate. So if it's been a while since you've thought about ATP, there it is again with the anhydride bonds. But this table is also showing some other high energy. So the more negative the delta G, the better. But here's the negative 32. It's negative 31.8, but we'll round to negative 32. So in order to make ATP, it's actually negative 32 kilojoules that's needed. So I would like you to commit that to memory. And you can see how, with going back to glycolysis, that we end up with a net gain of 2 ATP. And we said that this is the idea of substrate level phosphorylation. So when we look at glycolysis and other fermentation, it's pretty much the substrate level phosphorylation that we're talking about. There's one notable exception I'll talk to you about, but usually when you think about fermentation, think about substrate level phosphorylation. So what does that really mean? Substrate. So we're talking about a substrate in a reaction pathway. So it's at the substrate level that a phosphorylation will happen. That's how to interpret that phrase. So it's at the substrate level that phosphorylation would happen. So it's being shown here when you have this 1,3-bisphosphoglycerate right there in the pathway. And you can see it has 2 phosphates. And as you work through this, you can see that it's going to react with ADP. The phosphate will get shifted to ADP to create ATP. And then one of the other product is 3-phosphoglycerate. All right, so that's the substrate level we're talking about. The substrate, 1,3-bisphosphoglycerate, that's the substrate. It's donating off a phosphate to ADP. So it's at the substrate level that ADP is getting phosphorylated. So when you hear phosphorylation, usually that phrase, when you hear that, what they're saying is ADP is getting phosphorylated. That's what we really care about. All right, ADP is getting phosphorylated to make ATP. So when you think about how microbes make ATP, and us, we do glycolysis too. This process is substrate level phosphorylation. And when you look at these different fermentation pathways, they all are pretty much the same in the sense that they don't get the carbon molecule that you start with doesn't get totally broken down to CO2. It's a partial breakdown, which isn't as good. All right, so if you can break it all the way down to CO2, that's your best, but this isn't. So fermentation, any fermentation pathway, it's a partial breakdown of the organic molecule you started with, and there's no net, or there's no final or terminal electron acceptor in that process. Like what we see with the electron transport chain. Fermentation means that there's no oxygen, so it's anoxic without oxygen. A or AN in front of a and science word means without. So no oxygen environments. And so the types of microbes that do fermentation on a fairly regular basis are facultative microbes. So facultative anaerobe. If you're a facultative anaerobe like E. coli, do you know what that means? Some of you might have had med micro, or maybe you got it in lab yet. Facultative. Generally, think of it like take it or leave it. Facultative, well, I don't know if that's the right word, but facultative, if oxygen's present, they'll use it if they're smart. If oxygen's not present, I'll just do something else. All right, so the absence of oxygen isn't a deal breaker for them. So that yes, they can use oxygen if it's there, if it's not there, they'll do something else. So facultative anaerobe, if I were a microbe, I'd want to be a facultative anaerobe, because then you get a lot of options on what to do for generating your ATP. If you're an anaerobe, that means you're poisoned, especially if you're a strict or obligate anaerobe, you're poisoned by oxygen. So then you could run fermentation as an option. So those are the types of microbes that can readily switch to fermentation pathways. In the end, you'd get some ATP, so at least you get something. And like I said, it's through a substrate level phosphorylation process where this is a fancier way of saying it here, but a high energy phosphate bond is transferred from some substrate to ADP. And of course, that will get you your ATP. Okay, well, then we said you don't have complete breakdown all the way to CO2. So what's going on there? So you get some kind of product, it's not going to be all CO2. And then the microbe says, yeah, I don't care about that, so they excrete it, that's not their goal. The reason they're running the reason they're running fermentation is it gets them substrate level phosphorylation happening, so they can get ATP. Once they get the product, they're like, ooh, that's waste, I don't want it, and they'll excrete it out. Now, we take advantage of that in the brewing industry, because we make yeast do this over and over and over and over. One of the fermentation products, of course, you're going for is alcohol, but the microbe just excretes that. That's a waste product. In fact, that just builds up and builds up and builds up. And if brewers don't know what they're doing, they'll poison the yeast by having too much alcohol surrounding the yeast. So it has to be a very precise process to get the ethanol off, and feeding the microbe a lot of sugar so that they'll continue to do this over and over and over. You do see this NAD idea going on here. This is really good, because the microbes don't have a lot of these molecules, so they have to keep generating and using, generating and using. They have a very small pool of that. And you can see the ATP. So substrate level. Again, this is just a generic where you see substrate C with the phosphate. So it's whatever C is, that's the substrate that is donating off the phosphate to ADP. So these are some examples of different fermentation reactions. And I'm just trying to show you the diversity. There's a lot of options. So again, it just depends, the microbes are opening their pantry door saying, what do I have to work with? And they'll cobble it all together into a fermentation pathway with the goal of, I want to get some ATP. The way the products just get excreted out. Okay, so it's that's the product is not what they're going for. So you don't see any kind of an electron transport chain system happening during fermentation. And it's substrate level phosphorylation in order for them to run this pathway to get ATP. They have to do it through the substrate level. So you only get 2 ATP, it's not great. However, like in the brewing industry, if you just feed that yeast a ton of glucose, they'll use, yes, they're going to use a lot of glucose, but they'll end up making enough ATP to survive and reproduce. So they have to compensate that low efficiency. They have to eat a lot in order to stay alive. Okay, so they're very hungry, if you want to think of it that way, because they don't get a lot of ATP per glucose. But, they excrete large quantities of the waste products. So the brewing industry, that's a pretty good payoff for applied microbiology. Okay, so when you look at fermentation pathways, there's some common themes that are running, no matter what pathway you're looking at. Basically, there's some rules that have to be met. You have to have a balance of all the oxidation and reduction reactions that are going as you work through substrates to products. And the electron numbers have to equal out substrate to products. So the microbes are in the process of, like I said, they're kind of cobbling together this pathway, and they're just trying to figure out, they're trying to play by the rules, and they're trying to figure out, how can I put this all together so I can do substrate level phosphorylation to make ATP? And so electrons removed from some donor, some substrate, they have to get put onto another molecule. They don't really care what, as long as it meets the rules of the game, and as long as they can get ATP made in the process. And that's really how we end up seeing all these different fermentation pathways. It really depends on where the microbe has adapted, what's in its pantry, what's around it in terms of what does it have available to use. And so we call this whole strategy redox balance, that they have to make sure that the electron number is equal, substrate to products. And so that's one reason why hydrogen gas is often actually made during fermentation. Because there's a lot of protons generally hanging around, and so they can give off some electrons to the proton. And they don't really care so much about the hydrogen gas. It's just they're doing it to meet the rules of the game. And in the process of them doing all of this, they're doing substrate level phosphorylation. Another product that's commonly made in fermentation is acetate. That's another common fermentation product. But again, they don't care about the acetate per se. They just excrete it out. It just helped them meet the rules of the game so that they could continue running that pathway and get their ATP. So humans have figured out a lot of this in terms of how to get microbes to run certain fermentation patterns. You know, obviously with the brewing industry, that's a great example, but there's all kinds of other ideas out there that if you get to know your microbe and know how, what it can do with its physiology, you can force it to do all kinds of interesting fermentation pathways if you feed it correctly. And if it has the necessary, you know, adaptations to deal with that kind of food. You know, we often talk about glucose as the sugar coming into glycolysis, but it, you know, microbes can break down all kinds of things in terms of running their different fermentation pathways. So it's not necessarily that it has to be glucose to kick off a fermentation. Lots of options. And you can see on the bottom here, as they're going through their pathways, you can see ATP is getting generated. And of course, pyruvate tends to be, talk about that a lot as the branch point or decision point for the microbe. And so we've taken a lot of this and made use of it in applied microbiology, where you can see, of course, the alcoholic beverages, but butter and cheese and yogurt, and industrial chemicals. A lot of those are still made through microbial fermentation pathways. So, and fermentation is big business, billion-dollar industries. So, except I don't think most pickle producers ferment their pickles anymore, unfortunately. If anyone knows of anyone selling pickles in the area that are truly fermented instead of just being bathed in acetic acid, let me know. But most pickles these days on the normal supermarket shelves, they're not truly fermented. They're just pickled, so to speak. Okay, we'll talk more about fermentation. And you'll have the option in lab of doing an extra credit fermentation exercise, which Lexi will talk to you more about. So fermentation, making food through fermentation is a lot of fun, and it's very healthy in general. Well, the ethanol, maybe not so much, but some of the other food that you can make, definitely very healthy. We'll talk more about that. However, now we're thinking about this one weird example that I'd like to, because of course with microbes, pretty much nothing surprises me anymore when I read something about how microbes can do a certain thing. It's like, of course, it's a microbe. You know, they're amazing in their ability to adapt and survive. So this is an interesting case where they were studying this microbe and in the lab, and they were doing some experiments, and they kept calculating the free energy of, you know, they knew what they were feeding the microbe, what nutrients, and they were able to figure out what the microbe was doing, so to speak, with that. So they were calculating the free energy, and it never got to -32. It was short of that. And yet the microbe not only survived, but slowly grew. So they're like, okay, what's going on here? It doesn't have enough energy to do that. So then they figured out this microbe was doing something sneaky. They were using some ion pumps that were, so the ions were either protons or sodium. And what they were noting is the reaction pathway the microbe was doing, even though it was fermentation, it was enough free energy to pump sodium or protons, but it wasn't enough to straight out give them ATP. But it was enough to pump protons across the membrane. And that is good because that's one step away from making ATP. If you can get a gradient forming across the membrane, whether it's protons or sodium, if you have the right transmembrane proteins, this is going to work. And so they diffuse back through a special ATP synthase, and we're able to get ATP that way. So this is what they were doing in the lab. They were putting in succinate. Of course, there's microbes were, in a water base, and then they fed the microbes succinate, and they could tell propionate was building up and bicarbonate, and the delta G was only negative 20.5. So you need negative 32. And so that was a head scratcher. And then they had to look more deeply at what was going on here. And so here it is. Here's succinate. And you can see the microbe said, well, if I can link it to pumping sodium across the membrane, get my sodium to build up, do this enough times, there's the, propionate product, get this going enough where I get a lot of sodium pumped across, and then this very special sodium ATP synthase. The sodium came through instead of the protons, although they know protons can do this too with the right microbe. And you still get ATP. It's just a little bit more complicated. So this is something that was very surprising to them because they always thought, okay, fermentation is substrate level, but this is a fermentation reaction. You have a carbon molecule that ends up not being totally broken down to CO2. There's no terminal oxygen involved. So yeah, this is fermentation. And then the microbe was sneaky and linked it to, in this case, sodium pumping to get a sodium gradient. So again, microbes are amazing. They can do all kinds of things. They can make scientists very confused, but usually we figure it out. Okay. Questions on this idea? Okay. So yes, glycolysis is very famous. However, there's lots of options for microbes, and it really just depends on their habitat and what they've adapted to. All right. I'm going to get into the next slide set if I possibly can. Ugh. All right. No. All right. So we just worked through some examples of substrate level phosphorylation for the most part, except for that funky outlier. Then figure B is trying to get across the point of another way that microbes can make ATP. And we do it in our mitochondria, but remember, they don't have organelles, so they use their cell membrane. So what they're trying to show here is a microbe cell membrane is energized, which what they're trying to say here is that a proton gradient has been established. You have protons building up, they've been pumped out of the cell and they're building up outside, less protons inside. So there's your proton gradient, and as the protons diffuse back through the ATP synthase, then you get your ATP. So that's oxidative phosphorylation, which remember we were saying we have to be careful about that because so many people equate oxidative with oxygen, and that's not necessarily the case with microbes. They don't always have to use oxygen. So in this case, when we looked at the electron transfer chain, the microbe is using oxygen, which means it's a smart microbe. If there's oxygen available and they have that ability, absolutely use oxygen. But sometimes microbes aren't able to do that. So they'll switch to something else besides oxygen. So we refer to it as anaerobic metabolism. Some people call it anaerobic respiration. And basically, when you look at it and say what's different, there's something else besides oxygen as the terminal electron acceptor. Okay. So the microbe, let's say it's E. coli, it's swimming around in some broth you gave it, and it's like, where's the oxygen? Where's the oxygen? I don't see any oxygen. And then it goes, darn, no oxygen, but that's okay. I'll survive. I can switch to anaerobic metabolism instead if there's some key other nutrients available. So can be an inorganic like oxygen. It can be something inorganic, or it can be an organic molecule. Microbes are great. They can have a lot of flexibility with this. You still have an electron transport chain process. You still have all that going. There'll be different players, especially toward the end there with a terminal acceptor. And you see that if you calculate the free energy of that and the ATP generated, you don't get as much as if it was oxygen. So aerobic respiration is your top pick if you can use oxygen. But if no oxygen is available and you're facultative, that's okay. Again, the end product is not what the microbe is going for. That's just a waste product. All they're doing is playing by the rules of the game and trying to get their ATP made. So they run their electron transport chain. They link that to proton pumping, which gets them their ATP. So if you're E. coli and you're swimming around in a broth and E. coli is like, oh, there's no oxygen in here, then if you're a smart E. coli, you don't have any oxygen, what might be another molecule you might want to consider tapping into based on this electron tower? Yes, thank you. Iron. So iron is next up from oxygen in this particular tower, Fe3 plus. So that would be a great option, because remember, the further the electrons fall, the more ATP you'll be able to make. So you want to substitute, if you don't have any oxygen, you want to pick something that's close to oxygen on this tower. And in this particular tower, that would leave you with iron, or NO3 minus is nitrate. That would be another option too. And actually, a lot of microbes, at least that have been studied by humans in the lab, seem to really like nitrate and they seem to have a lot of it around them. So that's a common replacement for oxygen when they want to do anaerobic respiration. And so this might be something that they do, where they'll stick nitrate on the end as the terminal electron acceptor. And once that gets the electron, it becomes nitrite, but they don't really care about that. That's not their goal. They're just trying to get that electron flow so they can link it to proton pumping. And then some microbes can keep going and going and going and going. So this is actually showing how you can get, in this case, nitrate all the way to gaseous dinitrogen. And so it's the microbes that have created our atmosphere. They were here first for billions of years, and so they created our atmosphere that has 80% dinitrogen. And then there's other microbes that take the dinitrogen and bring it back into these other forms. We'll talk about that. So all of these last three are gases and can get released to the atmosphere. You'll be doing some lab experiments. You'll be testing this idea to see what microbes, if they can use nitrate as a terminal electron acceptor, and then how far can they take it? And some microbes stop right here at nitrite, and that's as far as they can go. They don't have any other enzymes to work it further. And other microbes can take it all the way. It just depends on what they've adapted to. So nitrate is often a favorite pick, although the iron would have worked too. And a lot of, especially around here, we have a lot of iron in our soil. So I imagine a lot of the microbes are using iron. So E. coli, for example, can do that first step, nitrate to nitrate. And then Pseudomonas is famous for taking it all the way to dinitrogen. And you need these microbes. You need to keep the nitrogen cycle on planet Earth going. And so I would like you to know this triangle, please. I would advocate you write it over and over and over, but basically the idea that some microbes can take dinitrogen from the atmosphere and we say they fix it, which means they've converted it into a form that organisms can use. And in this case, if you think about a lot of the microbes that hang out with plants can't act on dinitrogen, only the associated bacteria can. And so then the bacteria convert it to ammonia and give that to the plant. Say, here you go. And the plant says thanks. And then there's some microbes that are hanging out and they can take ammonia and act on it through what they call nitrification to get you nitrate or nitrite. And if I were a grower, which, when I'm growing plants, I definitely I would want this kind of fixation going on in my soil, and I want nitrification going on in my soil. So that aspect of the triangle, I would push. I would try to promote that. I would not be promoting this last phase of the triangle. If I was a farmer or grower of plants, I would not want denitrification going on in my crop fields. Because nitrate is the preferred form of nitrogen that plants love to take up from the soil. And so as the microbes are doing this, there'll be some nitrate. The plants say, thanks, I'll take that up. You don't want microbes acting on the nitrate and converting it to dinitrogen because plants can't touch dinitrogen. It's lost to them. They have no enzyme that can act on it, which is really sad. We have 80% in the atmosphere and they can't touch it. So knowing your microbes, if you are, working with plants, you need to know your microbes and you can engineer them to do what you want them to do as they coexist with your microbes. Side by side, on the left, we have aerobic respiration, which means oxygen sitting at the end as the terminal electron acceptor. On the right, you look for the terminal electron acceptor and it's not oxygen, it's nitrate. So by definition, that is anaerobic respiration or anaerobic metabolism on the right. But you still see there's an electron transport chain process. There's different players, especially since cytochrome O is devoted to working with oxygen, whereas nitrate reductase is devoted to nitrate. So some different players in the game, but it's the same idea. Let's get an electron transport chain process going, and with that electron flow, you get proton pumping. How many protons are showing up on the left there? How many? Add them up. 5. How many on the right? 4. So the artist is trying to show you don't get as good of a proton pumping going on here, which means you get less ATP. So again, oxygen is the best electron acceptor, and if you have oxygen sitting at the end, it'll generate the most ATP. Anything else, you still get decent ATP, but not as much. Does that make sense? Okay. So you'll be testing this in lab soon. But again, if you think about it, like what I was saying, because plants love nitrate and you want to keep that in the soil, you don't want this going all the way past. You don't want that. Not in your soil. Yes, we have to have recycling and get dinitrogen back into our atmosphere, but not in your soil where your plants are, if you're trying to make a profit. All right, that's it for today. We'll wrap this up on Wednesday. Then I think the next topic we're getting into is microbial growth.
Yeah.